my name is Jessica Fitzpatrick, and today I will be teaching you how to extract DNA from a strawberry. So the objectives today are first to summarize the steps in extracting DNA from a strawberry. Then you will learn the purpose of each material used in extracting the strawberry DNA, and I will teach you the purpose of using this technique. So you can use this technique on various fruits, vegetables, animal cells, and even human cells. I chose the strawberry just because that often yields a lot of DNA. You might have more difficulty if you used a more watery fruit like a grape or watermelon, because all the excess water will not give you as much DNA in the end. And you can use this exact technique on human cells, but it's just difficult because you often need about half a cup of cell material, and getting a half cup of human material might be kind of hard to do, so that's why we will be using a strawberry today. So the materials you have in front of you are 1 strawberry, 5 milliliters of chilled isopropyl alcohol, 2 milliliters of dish soap, 1/8 teaspoon of salt, a Ziploc bag, a coffee filter, 18 milliliters of water, 1 test tube, 1 funnel, and a cotton swab. The cool part about this technique is that all the materials you have in front of you are household items, and they're all very safe and easily obtained. The only real danger here is if soap or salt or alcohol got in your eyes, that might be uncomfortable, but you could just flush your eyes with water for about 5 minutes and it's okay. So it's a very easy technique, and all the materials are very convenient to get. So step 1 I already did for you. That was making the extraction solution, and you will find that in your Ziploc bag. In your Ziploc bag are the 18 milliliters of water, 2 milliliters of dish soap, and 1/8 teaspoon of salt. The purpose of the dish soap is to dissolve the cell membranes of the strawberry cells, and the purpose of the salt is to break the protein chains, and the water is just a solvent for all those materials to mix around together. So I already did that part for you, and I mixed around the solution, and the bags are a little sticky because it leaked through the bags, but yes, it should work. Step 2 is adding the strawberry to the solution, so this is where you come in. So go ahead and put your strawberry into your bag, and just squish it up until I get to the next step that you need it. Can you please squish this up? Thank you. Okay. So what you just did is you added the source of DNA, and by mashing it up for about 5 minutes, you're allowing it to come in contact with all of the soap and the salt so that it can start liberating the DNA, and you'll be able to see it in the last step. Step 3 is filtering the solution. So I'm going to let you guys squish that up for a little bit just to make sure you can really get rid of the large chunks. But this part's going to be, you might want to use your neighbor for this. That's probably good. Thank you. So you might want to use your neighbor for this because it can be a little hard to hold. But you're going to hold your test tube like this. Set your funnel up like this. Some of the funnel ends got a little fat, so they might be difficult to squeeze in there. So you set your funnel up, hold it into the end of the test tube, and you're going to just set your coffee filter into the end of the funnel. Try not to rip it, they're very thin. So once you have it like that, this is where your neighbor might come into play. You're going to open the bag and just pour enough into the filter, just a little bit at a time, and you're going to help it filter through by squeezing the coffee filter very gently because they rip very easily, and you don't want any of the large plant debris to get into your test tube. And do this until your test tube is about 2/3 full. Yeah, I know this part's a little hard. It's okay if you can't do it. I know you're by yourself. But yeah, so just fill it up until you have a decent amount, about 1/2 to 2/3 full. And once you have that, that should be pretty good. Does anybody need any help? Here, I can assist. Thank can just pour it in. Yeah, a little more. Okay, that's good. And then just gently squeeze it till you get however much that puts in. Okay. So the purpose of that step was to get rid of the large plant debris, because if you have huge chunks of strawberry in the actual test tube at the end, this could hinder your ability. Oh, I think you filled it a little too much. That's good. It might hinder your ability to see the DNA at the very end. So, is everybody ready to move on to the next step? Okay, I think so. Not the last step. The next step is adding the isopropyl alcohol. You'll find your alcohol in a small plastic container. And the purpose of this is to separate the DNA from the rest of the extraction solution and the leftover plant debris, because the DNA is insoluble in the isopropyl alcohol, so once you put it in, it'll actually separate out rather than dissolving in, and this is when you'll be able to see it. So you can use your funnel for this, or if you can just carefully pour it in, that will work too. But add all the contents from your plastic container into the test tube. Okay. Did anybody have any trouble with that? Okay, so what you should be seeing right now is the bubbles. They're going to look like stringy bubbles. That's actually the strawberry DNA. If you don't see it, it could just be because the solutions got very messed up over this past week because they kept leaking through the bags. So you may not have had all the components necessary to break up the DNA. But if you see the stringy bubbles, that is what you're looking at, the strawberry DNA. And what you can do now is you use... Oh, okay, that is it. You use your cotton swab, and I mean you can already see it if you see the stringy bubbles, but you can use your cotton swab to kind of pull it up, and it'll just look like a thick string of bubbles. That is strawberry DNA. And if anyone does not see it, I have some that I prepared, and please pass around your samples to the back of the room so that they can see it as well if you had a good one. Here. I will let you guys see this one.
We tried.
Okay. So you just learned how to extract strawberry DNA. Now, why would you ever do this? Well, if you were in a laboratory setting, that is when this is most applicable. Otherwise, this would just be a fun activity maybe for a science class. But if you're ever in a laboratory, then you can use restriction enzymes to isolate a certain part of the DNA. Then you could use PCR to amplify that gene, which means to make many copies of it, and then you'd have a lot of copies of that one gene. Maybe you could sequence the DNA if you wanted to look at what was going on in there. But this would not be exactly how scientists would do it, because this is not done in a pure sterile environment, but this is just giving you the gist of how each of the components worked. And extraction of DNA is actually usually one of the first steps in diagnosing a disease, so you kind of learn how they would actually get the gene out to look at that. So let's review. The first step was making the extraction solution. Can anyone tell me what was in the extraction solution? It was what was in the Ziploc bag. Does anyone remember what was in that?
Soap and water.
And salt. Yes. Do you remember the purpose of the salt and the soap? Okay. Does anybody remember?
Yes.
So the salt was to break down the phosphate chains?
The protein chains, close. Yes. And does anyone know what the purpose of the salt was? Or wait, we just got that. The soap. Yes.
Break down the cell membrane.
Yes, correct. And step 2 is adding the strawberry, which was your source of DNA. Step 3 was using the filter to get rid of the large plant debris. And step 4 was adding the isopropyl alcohol. What was the purpose of adding isopropyl alcohol? That was the last step. Yes?
To allow the DNA to separate.
Yes, because it was insoluble in the alcohol. So that was the steps and the purposes of the materials. And as as I said before, the purpose of extracting DNA would really just be in a laboratory setting, or if you really just, you could use this on, like I said before, various plants, vegetables, other cells, and you could compare the yields of the DNA if you ever wanted to, but really it would be in a laboratory setting or a medical setting for diagnosing a disease. And here are my references. Does anybody have any questions? All right, thank you. And you can just give me the test tubes and I'll just throw away the rest of it. Yeah, if you could empty the contents of the test tube into the plastic cup, that would be helpful.